Hello, and welcome to the next in our series of Plante Presents webinars. Today, we have two speakers, Magdalena Bezanilla and Joke De Jaeger-Braet. Our webinar is hosted by Adrienne Roeder, and moderator is Vijaya Battala. We are hosting this webinar to celebrate the publication of the January 2022 issue of the Plant Cell, which is a focus issue on the topic of cell biology. This focus issue was edited by Dolph Viegers, Magdalena Bezanilla, Liwen Zhang, Adrienne Roeder, and myself, Mary Williams. The issue is now online, and I encourage you to visit the website and look at all the great articles in the issue. That's it. We will be posting a recording of this webinar on our YouTube channel. You can also find the webinar that we ran 2 weeks ago, also featuring authors whose work appears in the focus issue. So visit Plante YouTube channel for lots of great science. We will also be using closed captioning. You can turn on the closed captioning at the bottom of your screen or turn it off. And we will be using the closed captioning in the YouTube video. We want to give a special thank you to ASPB members whose membership allows us to run this series. There's a financial support from the ASPB members, and we encourage you, if you're not already a member, to join the society. There's a special code, presents 10, that gives you a 10% discount on your membership dues. I encourage you to put questions for the speakers into the questions box. If you have technical problems, please feel free to let me know at mwilliams@aspb.org or through the chat box. And if you're having connectivity trouble, try checking your internet speed or dialing in. And now I would like to turn this over to Adrienne, who will be introducing the speakers. Oh, shoot, I've got to, there we go. Thank you, Adrienne.
Thank you, Mary. I'd like to introduce this focus issue a little bit more and tell a bit about how it came to be. It's been 33 years since the first issue of the Plant Cell in January 1989. And so we thought it was time to come back and reflect on the title of the journal, The Plant Cell. And so we're kind of going back to the roots of focus on plant cell biology. And as you all know, Plant Cell now publishes a wide range of plant biology articles, but we thought it'd be a good time to look at plant cell biology. This issue includes 11 research papers, and today you're going to be hearing talks by 2 of the authors, so I'll leave the science to them. But there's also 11 reviews, and I was thinking it'd be fun to show you a little preview of some of those reviews to get your interest in them. So we have 3 really special reviews that are vignette style reviews. So what we did is we asked a bunch of experts in the field to write small sections on areas of their expertise. The first of these is a glossary of plant cell structures. And it's more than just a glossary. It has current insights and future questions. And it has gorgeous images of the various parts of the plant cell. So I really encourage you to look at this. The second one is going back to some of the leaders and founders of the plant biology, cell biology field. And it has their personal reflections on how their science came into being through the years. And so it's really interesting. And the third one is a look forward. And we asked 15 experts to think about what are the most compelling questions to them in the field of plant cell biology and write about them. And so it's 15 open questions to stimulate your interest in various parts of plant cell biology. There's also a Plante blog where you can add your own questions. So I encourage you to do that. And then there are more traditional reviews covering the range of plant cell biology. And we hope that this will actually become a collection that people use as the go-to collection to understand the current state of plant cell biology. So there's one from Ying Gu and Carolyn Rasmussen on the cell wall. There's one from Liz Haswell's lab on mechanobiology. There's a review from Eugenia Rusinova on membrane trafficking. A review from Dolph Weiger's lab on polarization of cells. There's a review from Robert Soblowski and Crisanto Gutierrez on cell division. There's a review from Keiko Tori's group on fate specification and morphogenesis. A review from Satoshi Namorato's group on looking at evolutionary cell biology. And a review on imaging and quantification by Yvonne Gialis' group. And so a really amazing set of reviews that I want to direct your attention to. I think that the last question is, how will plant cell biology advance in the next 33 years? And that's really up to all of you. How are we going to take this field forward? It's a really exciting time. I'd like to conclude by thanking Mary for her organization of this webinar and the whole issue. It's, it's really up to her credit that this came off so beautifully. And now I'd like to turn it over to Dr. Vijaya Battala, who's a postdoc in my lab. She did her graduate work with Upal Math, looking at the role of TCP transcription factors in trichome development. And now she's working on giant cells in my lab. So Vijaya, take it away.
Thank you, Adrienne and Mary. Welcome, everyone. Today, it's my pleasure to introduce Professor Magdalena Bezanilla. She is a professor of biology at Dartmouth College, New Hampshire. She did her postdoctoral fellowship at Washington University, St. Louis, and received her PhD in cell biology from Johns Hopkins University, and her undergraduate degree in physics from University of California, Santa Barbara. Her lab focuses on polarized cell growth in plants, and for this, she uses moss Fiscal patents as a model system. Throughout her career, she has developed a wide range of tools in genome editing, gene silencing, and imaging. And the aim of her research is to identify the molecular mechanisms that link the cytoskeleton and vesicle trafficking to cell polarity. She has also recorded her talks on iBiology. If you're interested, go and visit that iBiology website. With that, I welcome Magdalena to share her slides.
Sorry, I had to unmute myself. Okay, great. Thank you. That was a beautiful introduction. And I'm really happy to be talking to you today about the story that was published in the special issue. So this story focused on this organelle, which is the endoplasmic reticulum, which is imaged here with a super resolution spinning disk confocal microscope. And I hope you can appreciate just how beautiful and intricate this organelle is. So my lab is really interested in understanding the underpinnings, the molecular underpinnings of tip growth. And as you are all familiar, tip growth is an important aspect of plant development. So in plants, seed plants, you have pollen tubes that require tip growth for fertilization and propagation of the next generation, but you also have root hairs that increase the surface area of the root structure, allowing for greater water and nutrient uptake. So my lab studies tip growth in the moss Fiscal Metrium patents because this is such a wonderful system for studying this particular type of growth. So what I'm showing you here is a movie of growing protonemal cells that basically grow the same way that root hairs and pollen tubes do. They do this by delivery of special material to the tip of the cell. But a benefit of this cell, of this tissue type, is that it actually does also do cell division. So while I'm not going to talk about cell division today, it's also a wonderful model system for discussing cell division. So I've called it P patents because it's undergone a name change. So now instead of Fiscal Metrium, we call it Fiscal Metrium, but really it's just still P patents, thankfully, and we can still call it Fisco. This is a beautiful image taken a long time ago by David Cove, one of the founders of moss biology. And it's an image of a plant growing on a petri dish. So for the reference, this is about the size of a US dime coin. So it's actually quite small. And it's great because we can grow it in the laboratory under very controlled conditions. So my draw to this organism is the fact that most of its tissues are a single cell layer thick. So I can just put the entire organism under the microscope and I get access to the cells immediately. So really excellent microscopy. Recently, we've been really revolutionizing how we work with this organism because of its very effective CRISPR-Cas9 genome editing. And then finally, there's just a great community with wonderful genomic resources. So I encourage you to look at that review that Adrienne just talked about, because they talked really nicely about some of the great developments for cell biology and Fiscal Metrium patents. Another really wonderful thing that I think is important for understanding a lot of what I'm going to tell you today is that we can regenerate whole plants from single cells. So here is an example of tissue that's been growing for a week after blending in the lab. We can scrape the tissue off of the plate, and then we can take the cell walls by digesting with cell wall digestion enzymes. We get a suspension of single cells. At that point, we can do things like transform with DNA, and then we can regenerate the whole plant. And this is an example of a plant 6 days after protoplasting. And you can see the initial protoplast here in the center, and you can see that the cells have grown out. So this is protonemal tissue. It's all tip growing tissue. We've imaged it with a fluorescence dissecting microscope, and you're actually seeing chalcophore staining of the cell walls. So it's a really wonderful system. It's fast. We can regenerate in a week, 2 weeks, and we get phenotypes very, very quickly after protoplast regeneration. So the focus of what I'm going to tell you about today starts from this long-standing history in my lab to try to understand tip growth. How does this process work? Fundamentally, we know that it requires polarized secretion. So we know that we have vesicles that contain interesting material. And what I mean by interesting is maybe the components there are more flexible, or there are enzymes that help to make the wall more flexible once those vesicles exocytose to the tip and secrete their contents. But once they do that, then this area of the wall is more flexible. And so you can allow the turgor pressure of the cell to push the more flexible area of the cell, and you get growth at that particular site. So the challenge to understand how this is working is to, what are the molecules that ensure that these particular vesicles are depositing here? How do they know to go there? And what's directing all of this? So that's really been the focus of my laboratory, and I started it a long time ago working on actin based on my interest from my PhD work. So this is a movie where you can see a tip growing cell with a marker that stains actin filaments. And hopefully you can appreciate that there's this dynamic focus of actin at the tip of the cell. And as that dynamic focus moves, the area that actually expands is basically predicted by where that focal point is. And if you just watch this movie replay here at the bottom, you'll notice as it moves here, it expands there. It's going to come down here, it'll expand. It's going to jog over there, it's going to expand. So really where the actin is predicting that focal point. We went on to show that here. Please focus on this top cell in the tube. There was no actin because we treated the cells with a drug that depolymerized the actin cytoskeleton, and then we washed that drug away. And now you can see that the actin comes back before the cell starts to expand. And that's just quantified here in the graph below. So the actin accumulates at the area before cell expansion occurs. So really actin is predicting this site. So for a number of years, we've been working on what makes sure that actin gets there. And recently we've made some insights on the small GTPases called the row of plants. Many people have worked on these master regulators of polarized expansion and just master regulators in general in the cell. And we've shown that they localize to the tip of the cell, and they're very important for polarized growth. We think that they're important for recruiting this very important family of actin nucleators that then serve to essentially generate that apical actin structure, which potentially works with things like myosin motors to help you bring the right vesicles to the correct place. So this is sort of where we're at, but we still really haven't gotten to the main question, which is what links the actin network to what's actually causing growth. And what's actually causing growth is vesicle secretion. And so we decided to take a genetic approach many years ago, and we did a loss of function screen, where essentially we asked the question, what happens if we remove, for example, myosin 11? Well, you get these tiny little plants that are composed of small spherical cells. So just to orient you, here is a control plant regenerated from a protoplast. You can see the beautiful filamentous tissue. This is now chlorophyll autofluorescence. And then here's a plant that's lacking myosin 11. Here's a plant that's lacking formin. Again, same sort of phenotype. And here's a plant that's lacking ROP. Very similar phenotype. So we reasoned if we could find plants that have this similar phenotype, but just with a forward genetics approach, maybe we would find connections that were not part of the cytoskeleton. And so we did that. And one of the first hits that we got was a SEC23 gene. SEC23 is a part of the COP2 complex. And this is a coat of proteins that assembles on ER-derived vesicles that are bound to the Golgi. So we were excited when we found that. And just as a little bit of background about COP2, SEC23, as I mentioned, is part of the inner coat of the COP2 complex. It heterodimerizes with SEC24, and then it recruits the outer coat. And then together, those things actually form the vesicle, and then the vesicle pinches off the ER and goes off to the Golgi. So SEC23 here is in yellow, and as you can see, it's got a number 7 next to it. And so when we discovered this about 10 years ago, we thought, oh my gosh, there's 7 genes. How are we going to tackle this? And it turns out its heterodimer partner also has 7 genes. So we just thought, you know, is this functional redundancy, or have these families expanded to have specific functions? And it took us a little while to get to this problem, but a talented graduate student, Ming-Ching Cheng, decided he wanted to get CRISPR to work. And so once we got that working, we decided we could tackle this problem. So let me tell you a little bit about the SEC23 genes. As I mentioned, there are 7 of them, and they are actually expressed in this tip-growing protonemal tissue, and 4 of them actually are expressed at pretty decent levels. Now, I grouped them into little groups based on sequence similarity. So there are pairs. F and G are more similar to each other. D and E are very similar, and B and C are very similar. But as you can see from groups 2, 3, and 4, you have representatives that are all very highly expressed. So we took a loss of function approach, and we used CRISPR-Cas9 to do this. And so what we discovered, what I'm showing you here again is a wild type plant. And we normalized the area of this wild type plant to 1. And now we're going to compare the sizes of these plants when we've mutated particular SEC23 genes. So one shocking thing was that if we get rid of A, B, C, F, and G, then only leave the class 3 genes, we actually see absolutely no phenotype in this protonemal tissue. In contrast, if we only get rid of SEC23D, which turns out to be the initial gene that we found many years ago in our RNAi screen, we actually see a very strong and significant phenotype. And this phenotype seems to be quite specific for protonemal tissue, for that tip-growing tissue. What I've circled here in this image is that these 3-week-old plants still form gametophores, and those gametophores are essentially the same size as wild-type gametophores, as well as the quintuple mutant gametophores. So there isn't really an effect except specifically on this tip-growing tissue. So we looked at the tip-growing, we then looked at the localization of SEC23D, and we did this by tagging the endogenous locus. And what you can see is that SEC23 forms these beautiful puncta along the ER network. So consistent with its role in participating at ER exit sites. Then we looked at the morphology of the endoplasmic reticulum in SEC23D null mutants. And what you can hopefully appreciate from these images is that SEC23D mutants form these aggregated clusters of the endoplasmic reticulum. And we were able to get a good super resolution view of these clusters, and it looks like these clusters are actually sort of aggregates of tubules of the ER. Then we wanted to see what was the impact on secretion. And what we noticed is that if we looked at a Golgi marker, we could see that in wild type, you essentially see mostly just the Golgi labeling, and you don't see the background of the cell. But in the SEC23D mutant, you see the Golgi labeling, but you see a very large high background in diffuse in the cell, indicating that this protein that's a Golgi resident protein is not efficiently getting to the Golgi. And we quantified that here. So you can see that you don't get efficient delivery of SEC20 of the Golgi resident protein in the SEC23D mutant, but it's actually fine in the quintuple mutant. So the quintuple mutant doesn't seem to be playing a role in delivering proteins to the Golgi. So then we looked at delivery to the plasma membrane. So we have a fluorescent protein that resides on the plasma membrane. And again, in the SEC23D mutant, you see a significant amount of that protein getting retained inside the cell. Interestingly, the quintuple mutant did have a phenotype here, and that's shown in this quantification. So the wild type, it delivers it fine. The SEC23D mutant has a big defect, and the SEC23 quintuple mutant has a defect, but not as strong as the D mutant. So we then were wondering, well, what are the differences between these SEC23 isoforms? And one thing that we thought to do was to analyze their localization. So again, we endogenously tagged 3 of the most highly expressing isoforms. So we chose D, which I showed you previously, and then B and G. And hopefully you can appreciate, in magenta is the SEC23 isoform, and in green you see the endoplasmic reticulum. Hopefully you can appreciate that B forms very small puncta, similar to D, but G forms very large puncta. And this is a very distinct localization. We quantified the size differences, and there is a very significant difference between the size of the G puncta compared to the size of the D and B puncta. Then we thought, well, one way to look at this was to analyze the co-localization between D and either B or G. So that is shown on this slide. So in the top, I'm showing you the co-localization of B in green and D in magenta. And you can see that there are instances where they're in the same place, instances where they're not in the same place, because of a large amount of both magenta and green dots. Strikingly, G and D are completely non-overlapping. So all G, remember, forms those larger puncta, and it doesn't overlap at all with D. So this is just quantification of the amount of co-localization using various different parameters. So this was really exciting because it is basically telling us that even though all these isoforms are still associating with the ER, they have functionally different consequences, and they seem to be associating to distinct sites on the endoplasmic reticulum. So what I've shown you is that B, D, and G localize to distinct ER domains, that secretion to the Golgi is required for tip growth, and this is predominantly mediated by SEC23D. And then that these other isoforms appear to be contributing to secretion of proteins to the plasma membrane. Whether that's through the canonical pathway or through other pathways remains to be investigated. So in the little bit of time that I have left, I wanted to just make another connection to a story about the endoplasmic reticulum from my lab, which is about a protein called Saber. Saber was discovered about 30 years ago in Arabidopsis. And it was thought to play a very important role in mediating polarity through the cytoskeleton. But what we discovered is that Saber actually localizes to the endoplasmic reticulum. So this is a beautiful turf microscopy image where you can see Saber along ER tubules. What's interesting is that it doesn't localize all over the endoplasmic reticulum. It actually localizes only to a small region of the ER. What I'm showing you here in this image is just the Saber channel, but these large chloroplasts bleed through in that channel. So it's not localizing to chloroplasts, it just happens that the bleed through of that channel shows us the chloroplasts. So this is just quantification showing that there is great co-localization with the endoplasmic reticulum and very poor co-localization with microtubules and middling with the actin. So Saber is an evolutionarily conserved protein found throughout eukaryotes. And so what you can see is that, in fact, it's been studied a lot in Arabidopsis. There are 2 genes, and there's also 1 gene in Drosophila. And what's really interesting is that it makes small organisms in every organism it's been studied. So it makes small Arabidopsis plants, and it makes small Drosophila. So it also makes small moss. And it's making small moss, as you can see, both in the protonemal tip growing tissue, but also in the diffusely growing gametophore tissue. So it's got a different phenotype from SEC23D, but it's still affecting cell expansion. And so here's just how it affects it. It actually stops growth for long periods of time, and then it'll resume growth. So the rate of growth of the tip growing cell is actually not any different when it's actively growing, but it is very different overall because it spends so much time not growing. And we know that the endoplasmic reticulum morphology is disrupted in this mutant. And recently we have clues as to what might be happening with the beautiful collaboration with Marissa Ategui at University of Wisconsin. What you can see here is the SEC23, is the Saber mutant. And the blue is the endoplasmic reticulum, green is the TGN, red is the TGN, green is the vacuole, and these white vesicles are just ridiculously large numbers of vesicles that you would never find in normal cells. And so we think there's a problem with membrane homeostasis. So just in summary of this, I'd like to tell you that Saber does localize to specific subregions of the ER and regulates cell expansion broadly, both tip growing and diffuse growing tissue. And it influences, we think, membrane homeostasis, and we're very affected. So with that, I'd like to thank you for your attention. I'd like to thank the folks in my lab who have done all of this work, and I appreciate, look forward to your questions. Hopefully that was okay in time.
Yeah, thank you, Magdalene. That was a beautiful talk. So I just remind the audience to put all of their questions in the Q&A chat box. So we do have one question. Does the fungal growth occur in a similar fashion by tip growth?
So fungal growth, especially things like Aspergillus and Ashbia gossypia, these fungi that are filamentous, they have something called the Spitzenkorper at the very tip of, and I'm saying that wrong, I know it's a German word, I'm saying it wrong, but anyway, they have this structure at the tip, and it is also heavily actin-based. And so I think many of the same components are there. And so I think there's a lot of parallels between the two systems.
Thank you. I have a question. So in your first part of your talk, when you're doing screening, you have shown mutants which lack myosin and all those. They really look like arrested kind of meristems. Are those arresting in meristematic stage? Are they following up, growing to some extent and then arresting?
That's a great question. Originally, when we did those RNAis, we thought that though they were sort of dead, that they would not be able to develop. But recently we've made a formin mutant plant that looks just like those RNAi plants. It's lacking almost every formin, and it continues to grow indefinitely. The ROP mutant, where we get rid of all the ROPs, grows indefinitely. There are some differences. So ROP, for example, can't develop gametophores. It just makes these round little cells that keep growing forever. Very slowly, but they keep growing forever. And then the formin mutant actually develops gametophores just fine. So it can actually go through development. So it's not really a meristem. I mean, these cells still, in the case of the formin mutants, they still have the identities, the correct identities.
Okay, thank you. We have some more questions from audience. Does Saber interact with the phosphoinositides?
So we have not looked at that, but I think the Drosophila group is looking to see if it's interacting specifically with certain types of lipids. I'm not sure that that was published in their last paper or not.
Thank you. Another question, does plant dwarfism has any relevance with Saber?
Oh, that's a great question. I don't know. I wonder if some of those dwarfs actually have mutations in Saber. I'm not sure that, yeah, that's a great question, and I don't know the answer.
So following up those questions, so does Saber have any known interacting partners?
We have not discovered any thus far. There are data from whole genome data from budding yeast, showing that there's interactions with a variety of different proteins, but those are all sort of like large scale experiments that need to be sort of validated. Yeah.
Yeah, thank you. Another question is, was the localization of the plasma membrane marker differentially affected in your mutants?
No. So the localization of the plasma membrane marker was the only thing that was affected was whether or not it got to the plasma membrane. So that was in the Sec23 story. And it just really was just whether or not it got to the membrane. So it was either stuck inside the cell or it was mostly on the membrane. But otherwise, we didn't see an effect like any other differences.
So another interesting question is, does Sec mutants influence cell division?
Great question. And no, not as far as we could tell. Cell division seemed to go on just fine in the Sec23D mutant. We looked at the timing of like fragmoplast expansion. We looked at the timing of mitosis, and all of those things seem fine. I mean, the cells do grow slower, and so, but the actual timing of mitosis and cell division didn't seem to be affected.
Yeah. Another question. Have you checked other biological processes involved in cell elongation, that is vacuole dynamics in different Sec mutants?
We did look at the vacuole with the Sec23 mutants. Unfortunately, we didn't have a great vacuole marker at the time because the vacuoles looked a little odd in the wild type. We have a supplemental figure in the paper showing that for the most part, vacuoles looked pretty normal, as normal as they did in the wild type. So but this is something that we definitely want to look at in the Saber mutant, because Saber mutants could be affected in their vacuoles.
Okay. Another question is, which type of ROP you found in the tip?
So there are 4 nearly identical ROP proteins in moss. And so it's not like a Arabidopsis that has a large number. And in fact, the moss ROPs are most similar to just one subset of ROPs in Arabidopsis and in seed plants. And so they're so similar to each other, they differ at most by 2 amino acids. So we really only looked at the localization of one, but we did that in the background of all the others deleted. And we validated that that was a fully functional protein because the plants actually continued to grow just normally. So you really only need one, and it can be tagged, but it has to be tagged internally with an internal tag.
Does gibberellic acid affect Saber behavior?
We have not looked at that.
Okay. Another question is, have you looked into any hormonal signal interactions in the mutants, both Sec and Saber? I think it follows up the.
No, we haven't, but these are great things to investigate.
Yeah, just let me check. Okay, I think we have one more final question. Have you checked other biological process involved in cell elongation? I think I have asked this one, sorry. Yeah, okay. I think that's it.
Thank you. Those are great questions. It's been really fun to share my work with you.
Thank you, Magdalena. Now we'll move on to our second speaker of this session. Okay. Today our second speaker is Joke De Jaeger-Braet. Hope I pronounced her name correctly. She is a molecular plant biologist currently finishing her PhD with Arx-Knitter at University of Hamburg. She obtained her bachelor's and master's degree from VIB Ghent. And today she'll be speaking to us about how climate change will affect the heredity. With that, I welcome Joke to share her slides.
You can already see my slides, right?
Yes.
Yes. Okay. So thank you for the kind introduction. I'm very happy to be take part of this this special webinar to celebrate the cell biology issue. And today I would like to show some of my work that I have been working on during my PhD studies and has been published in this issue. So let's move on. And with this, I would like to start introducing you to a topic that you all know very well, which is global warming, leading to different weather extremes And for plants, of course, this leads to high exposure to, for example, high temperatures. And for agriculture, this unfortunately leads to high yield losses. So there's a clearly an urgent need to understand temperature stress on yield-related traits like reproduction. And one of the essential processes for sexual reproduction and fertility is meiosis. It's a specialized cell division by which DNA replication is followed by 2 rounds of cell division, meiosis 1 and meiosis 2, leading to the production of 4 haploid cells and the reduction of the DNA content by half. Prophase itself can be subdivided into these stages, and is very important for genetic diversity, as the meiotic recombination pathway takes place during prophase. And it is initiated through the formation of double-strand breaks, which will then get repaired through strand invasion into the homologous chromosomes, and this will eventually lead to the formation of crossovers. And these are important for the further new assortment of the allelic combination and of course for genetic diversity. Another feature important during prophase is synapses. It's important for the very tight connection of the homologous chromosomes. And if we zoom in here, so also important for my talk later is AZ1, one of the main components of the chromosome axis together with AZ3. And ZIP1, which is a transverse filament, which is part of the synaptominal complex, which is formed during synapsis takes place. And due to its importance, it's not surprising that meiosis and its effect, or the effect of temperature on meiosis, has already been studied. And what we know already so far is that the microtubular cytoskeleton is affected by high temperatures, also low temperatures. We also know that recombination clearly is affected. Even at higher extremes, also synapsis is compromised. And we also already know that the progression of meiosis is also depending on the environmental conditions. And where these 4 aspects, they differ in the effects upon the different heat applied or heat stress. And I was further interested in the precise effect of high temperature on the meiotic progression in Arabidopsis thaliana, where I really wanted to look at the dynamics of meiosis at high temperature using live cell imaging. And quickly move this over. And to quickly show you or to remind everyone where we found myocytes, so the cells undergoing meiosis. Inside the young flower buds, you can find the male or the anthers, and these contain the male myocytes. And using confocal microscopy, we can easily look inside those anthers. And here the myocytes are located inside, clearly surrounded by several cell layers like the epidermis and the tapetum cell. And this might make you wonder, and also made us wonder, how fast do they really perceive a temperature shift, as they are so well embedded by and hidden by the plant. And for this, we made use of a study where they identified CDKA1 kinase being localized and recruited to stress granules upon high temperatures, here an example in roots. And I also made use of this CDKA1 reporter and had a look in myocytes. So first 21 degrees, which is our standard growth conditions of Arabidopsis, then 30 degrees, and an even higher temperature, 34 degrees. So the localization of CDKA1 in meiosis has been very well studied and characterized already, and has changing localization of nucleoplasmic and cytoplasmic localization. And here, in addition, you can see a microtubule reporter in magenta for the staging of the meiotic division. If we applied high temperature to these different meiotic stages, At 30 degrees, we could already see within 15 minutes several cells where CDKA1 is localizing or recruiting to these granules. And at 34 degrees, the cellular response was even higher, where we could see from Parkinson stage onwards, all cells we observed had CDKA1 forming these or recruited to these stress granules, as you can also see here from the quantification. And you might have already noticed that these early meiotic phases, their CDKA1 is not localizing to stress granules, or at least we don't observe them. But although given that these later stages show such a clear cellular response, we still assumed that also the early phases perceive the heat shift within a similar time. So with this experiment, we can clearly show that myocytes perceive heat stress and show a cellular response within 15 minutes. And as a little side observation, we see that CDKA1 is recruited to stress granules only in a stage-specific manner in meiosis. So now we can move on to live cell imaging, and quickly reminder how we perform live cell imaging in our lab. So the young flower buds with one sepal removed are directly embedded like this into medium and submerged with water, and were observed up to 48 hours with a time interval of about 10 minutes. And this gives rise to a movie like this, where here you can see the microtubules in magenta and autofluorescence in blue. A previous colleague from me in the lab, she described the meiotic division using different cellular features like the microtubulary states. And as you can see, these 7 states, they are very clearly distinguishable from one another. And we also thought it might be very interesting and useful to use the microtubulary states for the dissection of the meiotic division at high temperatures. But of course, we need to be first sure that we could really observe these different microtubulary states at the high temperatures. And I was interested in the following conditions. So again, 21 as our control, then both 2 heat shock treatments, 30 degrees and 34 degrees, where we applied a high temperature from the first microtubulary state onwards and followed the meiotic division at a high temperature. And on the other hand, also a long-term treatment where we already transferred the plants around bolting time to 30 degrees and around 1 week later, so it was constant 30 degrees, we observed the meiotic division. So if we have a look at these 3 different heat conditions, you can clearly see that the meiotic progression is proceeding. And besides some quantitative changes, like the spindle that we also see at 34 degrees, we were still able to clearly distinguish these 7 states, meaning that we can really use the microtubulary states for the dissection of the meiotic division at high temperatures. So let's first have a look at the total duration. So for the estimate of the total duration, we considered from late leptotene till the end of anaphase 2. At 21 degrees, we calculated total duration of about 1,270 minutes, which is about 21 hours, here together with the 95% confidence interval. And at heat shock 30 degrees, the total duration was further reduced up to 16 hours. And in heat shock 34 and long-term 30, the total duration was about 18 hours. So this was consistent with what has been reported before based on fixed material. But now we can go deeper and we can really have a look at the different microtubulary states separately. How do they behave at high temperatures? So there are several states that indeed, as you can see here, progress faster at high temperatures, where you can already appreciate some small differences between the stages and the different heat conditions tested. In addition, there are also states that seem to be less sensitive to heat, where the duration is not affected so much or by the temperature. But surprisingly, at 34 degrees, we saw a very clear elongation of Pachytene diakinesis, which was an elongation of about 7 hours. So with this, besides the fact that indeed the meiotic progression is faster at high temperature, we now could clearly show that not all meiotic stages respond equally to an increase in temperature. And we further found an elongation of Pachytene diakinesis at 34 degrees. And this made us also very curious to really find out what is going on and what is really driving the prolongation of late prophase. And one of the characteristics or important features for Pachytene is that cells reach a fully synapsed state. And this is one of the first features we then also checked. And in normal conditions, ZIP1 starts to be loaded at the chromosome axis from early zygotine onwards, which will then start to form into longer linear structures. And in Pachytene reaches a fully synapsed state, meaning highlighting the synapsed homologues. Now at 34 degrees, ZIP1 starts to be localized at the chromosome axis, but seem to fail to form these very long linear stretches. And in Pachytene, we could never observe really fully synapsed states. So it's clearly already that the problem of the elongation of Pachytene diakinesis is already there before. So to further, okay, so indeed I can show that the loading of ZIP1 is initiated but fails to continue. So to further narrow down really where the problem comes from or which part is really sensitive, we compared an early heat shock, so from the first microtubulary state onwards, with a different set of experiments where we applied high temperature from the second microtubulary state onwards. And we calculated the duration of Pachytene diakinesis. And surprisingly, what we see is that we lose the elongation of Pachytene diakinesis when we apply later the heat. And this clearly shows that it's processes during early prophase that are heat sensitive and then will cause the elongation of Pachytene diakinesis afterwards. And one of the prominent processes happening during early prophase is recombination. So we were, of course, wondering how is recombination affected at our heat condition. And for this, I performed cell spreads using DAPI staining to look at the DNA. And in normal conditions in Pachytene, chromosomes reach a fully synapsed or paired state, after which they will further condense into the formation of 5 p valents, which will align nicely at the metaphase plate in metaphase 1. Now at 34 degrees, Pachytene we could never really observe or found fully paired chromosomes, clearly already showing pairing problems. And in addition, we mainly found 10 univalents in diakinesis, with in addition some chromosome bridges between the univalents. So it's very clear that high temperature causes defects in recombination and synapsis, also confirming or consistent with what has been already reported before. Now, to further evaluate what is really, or to which degree the recombination is causing the elongation of Pachytene diakinesis, I made use of recombination mutants along the recombination pathway. So first, SPO11, which is important for the double strand break formation. Then DMC1, important for the strand invasion into the homologue, and MASH4, which is important for crossover formation. Now, first we have a look at these recombination mutants at 21 degrees. The duration of Pachytene diakinesis seems to be not different from in wild type situations. So this clearly shows that the loss of recombination itself is not causing this elongation of Pachytene diakinesis. So how do these mutants behave at 34 degrees? What's the duration of Pachytene diakinesis in these mutants? So as you can see here, we do see a slight increase in duration of Pachytene diakinesis, but clearly not to the level as we see in wild type. So this shows, besides that we now already know that the loss of recombination itself is not causing the elongation, the elongation of pachytene kinesis is clearly recombination dependent. But as we still see a little increase in duration, we know that, at the moment we don't know what is really causing it, but there is still a recombination independent factor playing there. And this elongation of pachytene kinesis always reminded us of the pachytene checkpoint that has been reported in yeast and animals. So quickly, what is the pachytene checkpoint in yeast and mammals? So it's an internal mechanism that triggers and senses problems during prophase and during recombination. And when these intermediate problems are sensed, the pachytene checkpoint will be activated, and this will cause or lead to cell cycle arrest. And in mammals, this often leads to apoptosis. And indeed, recombination mutants in yeast and animals, they arrest at pachytene stages, and clearly showing the presence of the pachytene checkpoint. Whereas we know from plants that the same recombination mutants, they do not pause at pachytene stage, they progress their meiotic division. And this always gave rise to the hypothesis that that plants would not have a pachytene checkpoint. But we also know that ATM has a very important role during plants for recombination. So we still went along and had a look at the ATM mutant and its duration of pachytene kinesis. And as you can see here, the duration of pachytene kinesis in ATM mutant again is a little bit increased from at high temperature or at 34 degrees compared to 21, but again clearly not the same elongation or the long elongation we see in wild type. So this piece of data clearly shows that it's recombination dependent. And in addition, we also now know or show that ATM clearly plays a role in the elongation of pachytene kinesis. And all these experiments together, they really made us believe that what we are looking at here is very likely to be a pachytene checkpoint. So plants seem to have a pachytene or a recombination checkpoint where the full recombination machinery seems to be needed to be present in order to trigger the checkpoint. And with this, I would like to come to a summary of what I've showed you today. So we show that myocytes show a clear cellular response to a high temperature through the formation of stress granules within 15 minutes. CDKA1 can be used as a sensor for heat stress perception and in a stage-specific manner in meiosis. We further confirmed that the meiotic progression is indeed faster at high temperature, but that not all meiotic stages are equally temperature sensitive. And we also pointed out that indeed high temperature causes recombination and synapses defects. And at 34 degrees, the duration of late prophase is recombination dependent. And it's the early steps in the recombination pathway that are heat sensitive. And with this, I would like to come to the end of my talk and would still like to thank a few more people. First of all, my lab members, and especially my boss, Arp, for the very nice time and great support. In addition, I also really want to point out Linda and Annika, which are great statisticians that worked with us together to build a robust statistical model for these data. And I would still like to thank one more time all the organizers of this webinar for all their great effort and put out this nice webinar. And I also would like to thank you all for your attention. Thank you.
Thank you, Yoka. That was a beautiful talk and beautiful images. I just want to remind audience to type their question in the Q&A box. So let's go to the questions. The first question is the asymmetric microtubule arrays at the late leptotin reflecting the position of bouquet?
So what is meant with asymmetric microtubule array? Maybe this person can.
Yeah.
Make this clear, or put a little comment on the chat.
I think he has put the under comment. I mean, the way the microtubules are all on one side.
So the so the half, I mean, we call this a half moon structure. Yeah. Okay. So it's already so it's so we know in Arabidopsis there's not really a bouquet formed. So there's more this concentration in density, but there's not really a bouquet formed. And the direction, I think there is no clear correlation yet between the microtubules and where the bouquet comes in other plant organisms.
Yeah, thank you. Another question is, can the change in membrane fluidity affect the duration of meiotic phase?
Good question. Yeah, I mean, what we know, of course, is indeed that the membrane fluidity is one of the early or one of the effects of heat. What we known is that, or from other people, that there is more callus deposition in later stages around the myocytes at high temperature. So this we know. So I guess this is kind of a mechanism from the cell, but whether they affect the progression, this I don't know at the moment. No.
Thank you. Another question is how conserved it could be in other plant species like rice and maize, considering those are normally growing under high temperature regimes.
This is a general, the increase of speed. I don't know if I can see the question. I see. Yeah, I think this is it's very well conserved. At least it's already shown in several species like wheat, for example, that indeed the meiotic progression is also faster at high temperature. They base themselves on fixed materials, so they didn't look into this very precise effects. So this should be checked still, of course. And then, of course, it depends on the growth condition, whether then it's also 34 in wheat. Of course, this might be a bit higher depending on their range of sensitivity in growth conditions. Yes.
Yeah, thank you. Another quick question is, does the effect of temperature is genotype dependent?
I mean, in general, yes. So what we see is we also checked other phases, of course, of the recombination mutants, and they also seem to be different already just in normal conditions. So of course, they affect itself already the meiotic progression. This has been also quantified and calculated and is also included in the paper, so can be looked at into more detail there. So yes, the effects, but I mean, you have to look at it a bit relative, of course. Yeah.
Thank you. I think we are getting to that last question of this session. The packaging checkpoint seems to be triggered by the heat stress at 34 degrees. Is this leading to apoptosis in meiocytes and thus causing the reduced pollen development associated with the heat stress?
No, so what they saw in mammals is that they really arrest in packaging, and then this triggers downstream targets up to that the cell undergoes apoptosis. And at 34 degrees, we see the meiotic division really finishing the whole division. And the fact that indeed, I mean, there is missegregation and so on, and this will eventually lead to pollen sterility. So it's not the checkpoint that is causing the sterility. This is, this is a different effect. Yeah.
Yeah, thank you. Still, there are many more interesting questions, but because of time limitations, we cannot go through all of the questions. And I want to thank you all the speakers today, and also like all the audience for attending this webinar. Thank you.
Thank you, speakers. Thank you, audience. Thank you, attendees. Thank you, ASPB members. And we will be posting this recording on YouTube in a few days. And look for an email. We have one coming up in February on a plant physiology focus issue on the topic of the plant cell atlas. So more exciting cell biology for you. Thank you, Adrienne. Thank you, Magdalena. Thank you, Vijaya. Thank you, Yoka. Thank you, everyone. Take care. Bye-bye.